All right, good morning, everyone. We're about to start the session. So thanks to everyone for coming up on that bright and sunny Saturday morning. I'm Fabrice, assistant professor at the University of Sherbrooke. Very happy to be chairing this session with my co-chair.
Hi, everyone. Good morning, and thank you again for coming out to the last early morning session for this conference. I'm Sarah Clark from the University of Colorado, and Fabrice and I had a lot of a good fun putting together this session. We're really excited about this space of looking at polymicrobial interactions. And I know, at least for me, in grad school, it was still all about we're just going to study one pathogen at a time, which has provided some really foundational knowledge and still continues to do so. But I'm excited to see the field move forward to thinking more about the complexity of these communities and how one pathogen is never really alone. And so we'll hear some stories about that throughout the session. So with that, I'd like to welcome our first speaker, Justin Liu. So come on up.
Good morning, everyone. My name is Justin Liu. I'm a current graduate student at Emory University in the lab of Dr. Joanna Goldberg. And the title of my talk is Experimentally Evolving a Staphylococcus aureus Cystic Fibrosis Isolate to Resist its Co-Isolated Pseudomonas aeruginosa. So starting off with things, I have no relations to disclose, sadly, but I do have some cool science to share. So as you all know, I work in Staph and Pseudomonas in the context of this disease, cystic fibrosis, a genetic disease that affects more than 120,000 people worldwide, and patients with CF are more susceptible to infections with pathogenic bacteria. The two bacteria I mentioned before, Staph aureus and Pseudomonas, are the most frequently isolated bacteria from respiratory samples of these patients. This is a graph from the CF Foundation 2023 annual report, where they look at the prevalence of microorganisms in the sputum samples of their cohort over the years. And you can see the two highest lines, green representing Staph aureus and blue representing Pseudomonas, are the most prevalent. So an interesting thing also to note is that even with the advent of new therapies such as ETIs, while they have reduced overall sputum accumulation in the airways, they do not completely eradicate Staph or Pseudomonas from these patients. This is a chart made by Morgan et al. In 2024, where they looked at positive cultures of both Staph represented by SA and Pseudomonas represented by PA about 3.6 years after start of treatment. Here we see that while the prevalence did decrease, it's still pretty positive or still pretty there. And also something to note is that bacterial respiratory infections remain the leading cause of morbidity in these patients even with these treatments. So looking more at Staph and Pseudomonas, while they're really frequently isolated individually, they can also be co-isolated from respiratory samples of these patients. This is a chart from an earlier report from the CF Foundation where they looked at the microbial diversity in their cohorts between the age groups. And you're able to see that really focusing in on the red or pink section that the cultures of both Staph and Pseudomonas really do become more prevalent in the patients when you see the increase in Pseudomonas as well. And these co-infections have been linked with worse clinical outcomes. And something also interesting to note is that while we do see a Pseudomonas increase in the prevalence in the older patient groups, it doesn't completely displace Staph aureus. This is a cartoon generated by Camus et al., where they looked at how we generally consider Staph and Pseudomonas interactions, where especially in the young groups, when they first acquire Staph and Pseudomonas, it's pretty competitive. But when you look at the older age groups, there's a lot more coexistence. And what really leads to the coexistence between Staph and Pseudomonas in CF patients isn't fully well understood. But when we really study Staph and Pseudomonas in the lab, especially when we compete them on agar plates, we see that lab strains of Staph and Pseudomonas, or we see lab strains of Pseudomonas greatly inhibit Staph. And what we first asked in the lab is is this also the same case of co-isolates, meaning that they're Staph and Pseudomonas isolates that were derived from the same sputum sample. And what we did is we screened 28 pairs of Staph and Pseudomonas co-isolates in this, there's our mouse, nice, in the screen on an agar plate, and they're able to see that in general, out of the 28 pairs, about 24 coexisted, meaning that the Staph levels and the Pseudomonas levels after co-culture were pretty equal, but they also identified some outliers. And one outlier was this pair SA46 and PA32. This is the results from the co-culture for that pair, where we have the SA46, we're showing you the CFUs of Staph recovery. So this is the SA46 recovered after co-culture of PA01 compared to Staph alone. And you can see while there is a decrease in SA46 recovery, when co-cultured with its co-isolated PA32, there's a more significant decrease observed, leading to the conclusion that, at least for this isolate pair, PA32 inhibits SA46 significantly more than the live strain PA01. And what we decided to do is we wanted to use SA46 and PA32 to really model the early staph and Pseudomonas isolates when they were first introduced. And I hypothesized that these two bacteria will identify new mechanisms and interactions between these bacteria. So when we first really wanted to study these bacteria, we wanted to do it in a more environment more clinically relevant, so we decided to use synthetic cystic fibrosis medium 2, or SCFM2, a media that mimics the artificial or landscape of C. sputum. And we competed both PA32 and SA46 together, as well as with laboratory strains of Pseudomonas PA01 and StatJ82. And we tested this because under laboratory conditions, in static co-culture, lab strains of Pseudomonas and staph have been shown to coexist. And this is just a quick setup where the Pseudomonas and staph were competing against each other at a 1-to-1 ratio in SCFM2 in these 4-wall chambers. And these are the results of the co-culture, starting with the lab strain J82. When co-culture with PA01, there wasn't really much of a decrease in J82 recovery compared to monocultures, which is consistent with what other groups have seen. But when co-culture with PA32, you see a significant decrease in J82 recovery. We see a similar trend with SA46, where while it is sensitive to PA1, it's greatly inhibited by PA32. Leading to the conclusion from at least this experiment that PA32 is hyperaggressive towards Staph aureus compared to PA1 in SCFM2. Next, we also wanted to check the supernatant, where we grew SA46 in the presence of PA32 and PA1 supernatant grown in SCFM2. We wanted to test this since secreted products from Pseudomonas have been shown to inhibit staph when grown in laboratory medias. This is a really quick and simple supernatant experiment where we extracted supernatant from 18-hour cultures of PA01 or PA32 to encapsulate most secreted things, especially things that are regulated by growth phase, and we grew SA46 in the presence of that supernatant. So going into the results first, this is SA46 grown with just filtered SCFM2. You can see that it grows just fine, but when it's grown with PA01 supernatant in the open circles and PA32 supernatant in the open squares, you can see that at 24 hours, the supernatant from PA32 inhibits SA46 growth more than PA01 supernatant. I keep pressing the 0. Leading to the results that SA46 is more sensitive to PA32 supernatant than PA01. So we have this one pair of Pseudomonas staph isolate PA32 and SA46, where it seems that SA46 is pretty hypersensitive to PA32. Next, we asked if we could identify potential mechanisms of tolerance or coexistence staph can develop through Pseudomonas using this pair. And we attempted this through an experimental evolution, where SA46 was competed with PA32 at a 10-to-1 ratio. This time, and this was all based on OD. They're competed statically for 24 hours before being transferred to this media called Bray Parker meeting that uses tellurite, a mineral, to select for staph and enrich while killing Pseudomonas. The selection happened for about 24 hours or 23.5, and then after that, a aliquot was passaged with fresh PA32. And this went on for about 15 passages, leading to about 79 observed staph generations. And something to note from this experimental evolution is that from the post-selection step to the next passage, there was about 2% that was transferred every time. But yeah, so going into the results from the experimental evolution, so after 15 passages, this is what it looked like. Starting with the black line, which represents SA46 alone. This was a SA46 population that went through the whole experimental evolution without ever being introduced to Pseudomonas. And then you can see that nothing really changed, but when looking at the SA46 population that was evolved to PA32, I did a total of 4 biological replicates, but I'm only showing you one. You can see that over time or over the passages, you see an increase of staph tolerance to PA32. And these graphs are all, so moving, specifically focusing on passage 14, where we see a first major bump in the staph tolerance to Pseudomonas. I picked single colonies and I competed them against PA32. So this is a single colony from the SA46 alone population. We can see that's still pretty sensitive to PA32. But from the evolved population, we were interestingly able to see both sensitive colonies and resistant colonies, leading to the conclusion that through experimental evolution, we were able to identify populations that had an increased tolerance to PA32. Tolerance was observed in the single colony level, and since we recovered both sensitive and resistant colonies, that there was heterogeneity in the population. So moving really forward and trying to figure out why this colony is resistant to PA32, we performed whole genome sequencing of this colony, which I'm going to refer to as ECR for the rest of the talk, and we identified mutations relative to SA46 using bioinformatics. And we were able to identify mutations in 2 genes. This one gene called GLTT, there was a frame shift, and a missense mutation in this gene called FAR-R. So GLTT encodes for an aspirated transport in Staph aureus. Interestingly, our lab has previously identified that mutations in GLTT alone was able to increase GA2 tolerance to PaO1 when competed in agar plates. And through a survey of clinical isolates, we found that mutations in GLTT are actually pretty rare. FAR, on the other hand, encodes for a regulator of the FAR-E fatty efflux pump. Some groups have seen that when you have missense mutations in FAR, it increases the export of fatty acids. And they also noted that mutations in FAR have been observed in Staph aureus isolates and are pretty prevalent in certain clades. So next we want to ask if mutations in both of the genes are required for tolerance or if it's mainly due to one, such as GLTT. So we individually complemented both GLTT and FAR from the SA46 wild type back into ECR on a plasmid, and we tested that against PA32. So these are the results of the co-culture. The main thing to note is that when we complement either the GLTT gene or the FAR gene, it restores sensitivity, leading to the conclusion that under these conditions in SAFM2, mutations in both these two genes are required for SA46 to have an increased tolerance to PA32. So in summary, starting with the first part where we talked about PA32, PA32 antagonizes its co-isolated SA46 greater than PA1 in SAFM2. Supernatant from PA32 led to a more sustained inhibition of SA46 compared to supernatant from PA01. And in future experiments, we'll aim to identify the differences in metabolites in these supernatants that may be responsible for the observed effects. And looking at SA46 tolerance mechanisms to PA32, we found that through experimental evolution, we found a population of SA46 that developed tolerance to PA32 by acquiring mutations in GLTT and FAR. Complementations of either GLTT or FAR restore sensitivity to PA32. And in future studies, we'll explore how these mutations in these genes confer tolerance to PA32. With that, thank you, everyone.
And I'm wondering if you've had the opportunity to look at clinical isolates to see if the mutations that you evolved show up in people when staff is contact.
Yeah, so we did do a previous screen with GLTT in a database of clinical isolates that range from different infections, and they say that while there are some mutations in GLTT observed, the frame shifts aren't really observed that much. We do have a collection of a lot of sequences, and I wanted to explore more FAR and see if that's really prevalent, because I think the previous studies have just really focused that on specifically infections from clonal complex 5, but then other clonal complexes that do infect patients. I want to see if that's also prevalent there.
I have a question from Barbara.
Hello. Yeah, Barbara Kahn, Münster, Germany. Hi. Very nice experimental evolution. So my question, maybe I didn't understand it correctly. So you co-isolated or you co-cultured Staph aureus and Pseudomonas aeruginosa together, right?
Yes.
Yeah. Okay. Did you also look for mutations in Pseudomonas?
That we did not, because I think when we did experimental evolution, we mainly wanted to introduce fresh Pseudomonas just to see how Staph is evolving for Pseudomonas rather than co-evolving together.
So you didn't co-culture them. So
Oh no, it did. It's just that I killed Pseudomonas off after each passage and then introduced fresh Pseudomonas.
Okay. Okay. Thank you.
Got two questions for you, Justin. That was a great talk. So do you know if these two mutations you found in Staph are mutually exclusive, or do they have to come together? Are they synergistic or antagonistic?
Yeah, so far all we really know is that the mutations are synergetic, because especially if we complement them individually, it restores sensitivity. And I also tested a GLTT mutation by itself, the one that was previously published, and that alone wasn't enough to confer resistance in SCFM2.
Okay, cool. And if you were to do the experiment so you've shown that beautiful plot from the CF registry where Staph is first and then Pseudomonas comes afterwards. So if you were to model this, so instead like growing Staph in SCFM2, then add the soup from your Pseudomonas isolate, would you see the same killing phenotype, or is there some type of protection? Or
I think if you pre-grow Staph at SCFM2, there might be a little more tolerance since you're able to establish these Staph like biofilm or something like that. But I think we would still see killing, especially when you co-culture with Pseudomonas.
question here.
This must be a silly question from a clinician, basically, but do those mutations that you found on Staph that confer more resistance to the Pseudomonas isolate have any effect on how they respond to host responses or antibiotics or anything like that? And that might be too early to know anyway. But
yeah, there is a possibility that the mutations in especially FAR, which is a fatty efflux pump, might confer resistance to the host or host offenses or as well as antibiotics. anything that really uses fatty efflux as a mechanism would be like pumped out. GOTT, we're not really too sure, I'd say.
Can I ask a question? So you had this interesting result when you picked the single colonies of some that were resistant and some that were sensitive. So do you know if it's beyond the heterogeneity, like how do those sensitive ones survive? Are they like benefiting from the ones that are resistant in some way?
Yes. That would be our idea, is that while not all the populations be resistant, if some of them are resistant, it may help overall. If it just is better at exporting fatty efflux plants, if those that can act as a sponge for pushing out all the fatty acids, then it could be a really good way to protect the rest of the population.
Cool.
More other questions? So let's please thank Justin for his talk. So our next speaker, our next speaker is Melissa McDaniel from, hope I don't get this wrong, Radboud University in Ireland.
All right. Hi, everyone. Thanks so much to the organizers for allowing me to be here and share this work with you all. Today, I'm going to be talking about a project where we've been looking at how Pseudomonas aeruginosa is able to kill non-tuberculous mycobacteria in the context of dual-species biofilms. As was mentioned, I'm a postdoc with Dr. Jacco van Ingen at Radboud University Medical Center in the Netherlands. But I also want to point out that this project actually started here in the States in the lab of Jessica Schofield at the University of Alabama at Birmingham. And I have no relationships to disclose related to this presentation. So the non-tuberculous mycobacteria are really what I'm going to focus on in this talk today. These are a group of very diverse bacteria that we often find in the environment. In terms of CF, we know that right now we're seeing NTM in about 12% of patients. And you might notice here that the median age at first infection is 27 years. So this is a sort of late stage colonizer and is considered sort of an emerging pathogen in the context of CF. The two most common species that we see in this patient population is going to be the Mycobacterium avium complex, which is the most common, followed by Mycobacterium abscessus. But we kind of give special attention to Mycobacterium abscessus because it in particular is associated with a very severe decline in lung function. I'm putting up this giant chart not for you to read, but just for you to appreciate that treatment regimens for the non-tuberculous mycobacteria are very complicated. They require a lot of antibiotics for a very long time and are very species specific. So you really need to know what you're working with to pick the regimens. These are at least 12 months long, usually 12 to 18 months, and are very poorly tolerated. And we know that despite having defined these regimens, that for Mycobacterium abscessus, at least subspecies abscessus, we still only have about a 35% cure rate, even when the prescribed antibiotic regimen is used, really highlighting that we need more effective therapies for this group of bacteria. And I wouldn't be in this session today if I wasn't going to talk about polymicrobial interactions. As we know, these are very important in the context of a CF lung, and I think up till this point, Pseudomonas has really kind of sat as the key player here, at least in what has been studied. And so many interactions with Pseudomonas have been described for a variety of bacteria, fungus, and viruses. And so what we know in the context of Pseudomonas and NTM is that about 25% of NTM infections are going to have a Pseudomonas co-infection. So we're seeing this fairly frequently. We also know that the presence of these organisms is actually negatively correlated in patients, but those that have both tend to have a more severe decline in lung function and an increased frequency of exacerbations. And for those of you who went to Nikki Lamoli's talk yesterday, maybe you can appreciate that oftentimes what happens when bacteria are antagonizing each other is an upregulation of virulence factors, and the host is ultimately who suffers. And so seeing this data, we had the hypothesis that Pseudomonas aeruginosa and Mycobacterium abscessus are going to have an antagonistic relationship. And because we're in CF, we started testing this very simply with just a dual species biofilm. So we took our Pseudomonas, our Mycobacterium abscessus, and just grew them together in rich media in a static biofilm in a 96-well plate. And we did this both for PaO1, sort of the normal lab isolate, but also for a clinical strain from a person with CF that is mucoid. And so what you can see here for the Pseudomonas, we're going to have our single species in a solid line and our dual species biofilm cultures in the dotted line. And the Pseudomonas did not care at all about the presence of abscessus, not a single change. But when we look on the abscessus side of this, we find that abscessus by itself is growing really well, but that when you put it with Pseudomonas, we get this very rapid decline in viable mycobacteria. And this might not look that impressive to people who work on fast-growing organisms, but 4-day clearance for an NTM is pretty impressive. We also looked at this for our clinical strain, and we find, again, no change to the Pseudomonas, but that we are getting rapid killing of our Mycobacterium abscessus, albeit a little bit less effective. And I mentioned I checked this in a biofilm context. So we also confirmed this via confocal microscopy, where you can see the Pseudomonas in green and the Mycobacteria in red, both happily making biofilms, single culture, and in dual species culture, you can see that we've really almost eradicated our Mycobacteria at the 72-hour time point. We then wanted to know if this is a contact-dependent mechanism or if this is a secreted factor by Pseudomonas. So we did a similar setup where we took Pseudomonas, we grew it as a 6-well biofilm for either 24, 48, or 72 hours, harvested our supernatant and sterile filtered it, and then used this as the growth medium to then seed Mycobacterium abscessus biofilms, evaluating the amount of death by colony-forming units. And what we find here is for both of our strains of Pseudomonas that supernatant is sufficient, so it is something secreted. And interestingly, this seems to get more extreme as we age those biofilms. So you do see death with the 24-hour biofilms, but it's really those 48 and 72-hour biofilms that are much more effective, which we see for both strains. And so we've been looking at biofilms up to this point, but I was also curious if this is something we see planktonically. And interestingly, the answer is no. So when we harvest Pseudomonas supernatant from a planktonic culture, this really has lost its killing ability, and we require Pseudomonas grown as a biofilm to potentiate this killing. So I started with Mycobacterium abscessus, partially because it's our fastest grower, and also because it's the one we know causes the most severe decline in lung function. But we were curious how this would hold up against other NTM species. So we tried a panel of rapid growers in both planktonic and biofilm grown cultures. We find that for abscessus, for Mycobacterium fortuitum, and for Chelonae, we again see killing both planktonically and in biofilm cultures with complete clearance by day 5. And then for our slow growers, including avium, intracellulara, and chimera, we again see complete clearance by day 5 in our planktonic cultures. And maybe unsurprising with the growth rate, we see a little bit of a lag in the killing with our slow growers, but are still seeing significant death with those ones as well. So now is maybe a good time to bring up that I am not the first person to study this phenomenon. So there are at least 2 papers out there who have studied this pretty in depth, one out of Daniel Wozniak and Luann Hall-Studley's groups, and one out of Kyle Rhodes' group. And in both cases, they found this inhibition, but were unable to really come up with a satisfying mechanism for this. And I now kind of benefit from the fact that they have looked at a lot of our more common Pseudomonas sort of systems to kill other bacteria. And so this indicated to me that I probably need to start with an untargeted identification approach. And so to do this, we did some solid phase extraction just to do class separation and narrow down a bit, followed by identification with liquid chromatography, mass spectrometry, and eventually we are going to follow up with exact molecular identification. But before we could get to this molecular work, we had a couple questions, and the first is if we're looking at proteomics or metabolomics. And so pretty simply, I just filtered this supernatant with a 3 kilodalton filter, which will take out most of your proteins, and we find that this makes absolutely no difference in killing, so we're probably looking at a small molecule inhibitor. We also needed to know the pH of the supernatant, and I'm not sure what I was expecting when I measured this, but I definitely was not expecting what I got, which was a pH of 8.75, which is incredibly high. We found this for both of our Pseudomonas isolates, and this really kicks up around 24 hours for both of them. And I saw this and I was like, oh great, there's our mechanism. It's not interesting. We probably can't change it in patients. It's just the high pH. But to test this, I took both the rich medium LB and raised it to a pH of 8.75. And you can see we got about half of the killing we'd expect. And then on the other end, we took our supernatant, lowered it back down to a pH of 7. And again, we lose about half of our killing. And so this really indicated to us that we need both of these factors, the high pH and something secreted in our supernatant to get our killing. We then moved on to our class separation, so I won't get into too much detail, but this is just C18 columns that allow us to separate our polar and our non-polar molecules. And what we find is that with our polar molecules, we get a little bit of killing at either pH, but not quite as effective as we would expect for the results we've had in the past. And then I'll call your attention over here to the non-polar fraction. where in our low pH we see basically no killing, but in our high pH we see very effective killing. And so this was the fraction that we decided to move forward with. We then wanted to separate a little bit more, and so we used the same sort of experimental setup, but with a little bit of a twist, which is that we can load our supernatant on at either a very basic pH or a very acidic pH, and this will allow us to determine if we have a basic molecule or an acidic molecule. So in our basic setup, our bases are not charged and will retain on the column, which we can then use. In our acidic medium, our acids are non-charged and will stay on the column to then be used. And what you can see over here is our raw supernatant is, of course, very effective. The methanol, our nonpolar fraction from our neutral supernatant, again, very effective, as well as the basic. But when we get to what was put on at an acidic pH, we've lost our activity, indicating to us that the bases flowed through, and so our effective molecule is a base. So that took us to at least knowing mostly what we are looking for when we run the mass spec, which helped us narrow down quite a bit. So we went ahead and ran this on our effective and ineffective fractions. And we started with about 55,000 features that came off of the mass spec, which is a very intimidating number of things to try to sort through. But thankfully, we had those different fractions that allowed us to filter quite a bit. So we first picked retention time. So we're expecting a base to stay on our setup quite a bit longer than an acid. We filtered for things that had high sample abundance, low variation between samples of our effective fractions. And then ultimately, we looked for a log two-fold change between the effective fractions and all of our controls that was greater than 1. And you can see our results over here with the effective fractions we're expecting in the biofilm portion and the less effective fractions being our planktonic and our growth controls. And ultimately, we have at this point 19 features that we're moving forward with. We have run those through the Pseudomonas aeruginosa metabolomics database. Some of them have hits, a lot of them do not. And I think this is going to require some confirmation, definitely tandem mass spec to identify these better. And then ultimately, we will move forward with those sort of isolated molecules to see their killing potential with NTM. Before I end, I wanted to bring these papers up again, just because we have some differences in the conclusions that we've come to. But I think that we have different growth media here, lots of different strains, and I think ultimately this is a good thing. So I think that, as we know, the Pseudomonas has a whole host of mechanisms by which it can kill other bacteria. I think we're probably also looking at multiple mechanisms by which it can antagonize NTM. And I think this is a strength. I think this means that we have a lot of different things that we can sort of work towards to develop effective anti-NTM strategies. And so kind of conclusions here. We know that co-culture of Pseudomonas and Mycobacterium abscessus and every other mycobacteria that I tried results in a rapid decline in viable mycobacteria. We feel that this phenomenon is contact independent but biofilm specific, and that this killing is mediated both by a raising in pH and a basic nonpolar molecule that we're still working on identifying. And ultimately, I think that there is likely a conserved pathway between NTM species that Pseudomonas can exploit for killing, and this to me is very exciting given how species-specific we currently have to be in our treatment regimens. And so with that, I'll acknowledge my lab over in the Netherlands, also Jessica's lab here in the States, and then the functional metabolomics lab at Radboud that helped us with all of the mass spec work. And then I am funded by a CF Foundation postdoctoral fellowship, which I am very thankful for. And so with that, I will take any questions.
Hi, Katie Wetzel from Georgia State. So the killing across different species of mycobacteria was pretty cool. I was curious, and maybe you said it in the beginning, what strain of M. abscessus you used for this experiment, and if you have tried other strains, other morphologies.
Yeah, so that one was just ATCC 19977, so kind of boring on the pick, which was partially because it's at least subspecies abscessus, so it's the one that we don't treat very well. I have tried this with the rough and smooth morphotypes separately. That's not in this work, but I can tell you it kills the smooth a little bit better than the rough, which is maybe unsurprising. We are currently picking isolates out of our sort of repository that have been isolated with or without Pseudomonas. So we're planning to do a host of clinical strains soon, which I think will be interesting.
Yes.
Hi, Bu Sang, University of Nevada, Las Vegas. I'm not sure if this thing's on.
It's on.
I can hear
Okay,
you.
cool. I was just wondering, and I think I just might have missed this. So in the plankton, when you grow Pseudomonas planktonically and then isolate the soup, is the pH also still super high?
Yeah, good question. It is.
Okay.
which I thought was really interesting, which I think also goes to show that it is something sort of synergizing with the pH and not pH alone. Because yeah, that was one of my biggest questions, was maybe that was just a biofilm pH change and that was what it was. But no, I got it high for both types.
Cool. Thanks.
Hi, Shoshana Jha from UCSF. This is extremely interesting, and it was very clearly presented. Thank you. So I actually am more in the non-CF space, and this is my first NACFC. It's awesome. And so we have a lot more NTM in the non-CF bronchiectasis, and we see in our registry that those that have Pseudomonas actually have a lower frequency of NTM, so this is very interesting. My question to you is, were the NTM corded, or were they single organism when you were growing them? How old was the culture before you added Pseudomonas? Because you know that the biofilm formation of the NTM would be different in the airway.
Yeah, so these were pretty fresh cultures, and I was using the supernatant as sort of the biofilm forming media. So these were 3 to 5 day cultures, and then I subculture for 24 hours as sort of like a fresh subculture, at least as far as you can get for mycobacteria. But I have actually tried this where I pre-formed mature abscessus biofilms and then put the supernatant on, and the killing is maybe, as you'd expect, less effective, but there is still killing. So it seems to work both ways. Yeah.
Hi, Brittany Fung from Cedars-Sinai. Great talk. I was just wondering if you could speak on whether you are expecting this factor to be a byproduct of biofilm formation or a actual response to NTM.
So I have tried this. The supernatant you saw was actually just Pseudomonas single species biofilms. And I have actually tried the single and dual species biofilm supernatant as the killing, and I see no difference there. So to me, I think it's just a byproduct of Pseudomonas biofilm formation, but not really a response to mycobacteria.
Hello, my name is Hiroaki Murano from UNC. I'm not familiar with bacterial biology, but my question is, have you ever tried this model on the human airway cultures?
Yes. So I have, and I don't see killing. I can go ahead and tell you, but the time scale on that is very different, right? We can only really keep those going for 6 hours before the Pseudomonas starts to overtake the cells. And even in vitro, I don't see killing by 6 hours.
So in that case, your model was non-CF culture or CF cultures?
So this one was just LB. I will say I have tried this in SCFM too, and I actually see a lot less killing in that context, which I think may be partially pH, because we know that's a more acidic environment. It might just be that Pseudomonas isn't hitting quite the growth phase for that. But I also think it explains some of the literature where in non-CF bronchiectasis in particular, these seem to be antagonistic for lung function decline, but in CF, that's like a little bit less clear. So it might be that actually the CF environment allows them to coexist a bit better.
Thank you.
And I have a follow-on question. My name is Heather Bean. That is sort of related to the prior question. Have you tested your bioactive fraction that kills NTM for toxicity to epithelial cells or to immune cells?
Good question. The answer is no. I can tell you I am anticipating that it will be toxic. So I am kind of under no illusion that Pseudomonas supernatant is going to do very well in the lungs of anybody. And I think what we're aiming for more so is looking for pathways in the NTM that then would be things we could target with more traditionally developed antimicrobials later, or maybe at least some altering of our active molecule to be safer. I haven't tried it, but I'm anticipating it would not go well, to be honest.
So we have a question in the chat. So do you think treatment of the airway with hypertonic saline would impact the killing by Pseudomonas on NTM?
I think it's very possible because you're, readjusting a lot of that environment. I will also, though, give the caveat that I'm showing this in vitro, and I think that they have the ability to kill each other, but I'm not sure how much evidence there is for direct killing in the lung. And so it might, I'm not sure how much I can speak on, if that's happening in the first place, and then if so, how much it would change.
I have a question for you. So in one of your slides at the beginning, you showed that co-culture with the clinical isolate of Pseudomonas and NTM, and then you could see that sometimes it would go up and then go down, go up. Could you comment on that?
Yes. So one of the things that I haven't talked about, but is big in the NTM field, is the viable but non-culturable state. And I'm saying killing here with the expectation that I think it is killing, but it might be that we're also having a sort of tolerance population where they're going into quiescence, bumping back up, bumping back down. And I will say, I also didn't mention this in the methods, but in those dual species biofilms, I'm feeding them every day with fresh media. So they are at some point kind of getting an influx of new nutrients. So they might be kind of popping up and then back down as it goes. Yeah.
Let's think. Oh, one last question.
Riquelme from Colombia. Just a quick question, just out of curiosity. What happened to the victim here? How NTM is dying? What is happening?
Yeah, we don't know. I have done RNA-seq experiments, and I maybe should have predicted this, but there's so little NTM there that we really weren't able to get a signal in that context to know how that was happening. So we do plan to do that either with hybridization for the transcripts from the NTM so we can get a better signal. I will say on the other half of this work that I'm not presenting today, we do actually see a benefit to the Pseudomonas biofilm when this happens. So we get a thicker Pseudomonas biofilm during co-culture. I know from experimentation that is PEL and eDNA based. With the eDNA result, my guess is that they're lysing, and that is being used by the Pseudomonas, but I have no great evidence for that. That's just a guess.
Great, thank you.
Yeah.
All right, let's thank Melissa for her talk.
Thank you.
Great. Next up, it's my pleasure to welcome Ed Swords. Come on up.
And I'd like to thank the organizers for giving me the opportunity to speak. Before I even get started, and this will be on the acknowledgement slide, but the hero of this story is a recent graduate from my laboratory, Caitlin Billiot, who did this work for her PhD thesis. Okay, I have no relationships to disclose. All right, Achromobacter is often mentioned as an emerging opportunistic pathogen in patients with CF and other debilitating airway conditions. It's a gram-negative, non-fermenting bacterium that's widespread in environmental sources, including potable water. It's highly resistant to most antibiotics, and typically it's observed in patients who are the sickest. So it colonizes patients. The correlation, clinical correlates indicate that observation of Achromobacter xylosoxanins is an indication of poor outcomes. Often it's seen in conjunction with Pseudomonas aeruginosa. Okay, so the first thing Caitlin wanted to ask was, do Achromobacter and Pseudomonas cooperate in vitro? And so this is just the simplest assay, doing a biofilm formation. In each case, we have transformed them to express fluor, and then in a static chamber vessel on an abiotic surface. And what I hope you can appreciate on the right is that they, when infected together, they form fairly dense polymicrobial biofilms containing that are pretty well interspersed with one another. Okay, so the next step that we went to is to ask, does Achromobacter form these aggregates on a more relevant surface, being the airway epithelial surface? We used immortalized CFPE epithelial cells cultured in an air liquid interface. This is kind of an acute infection because, as Dr. McDaniel mentioned, there's a limitation of the viability of the epithelial cells. They start to die really around 6-7 hours. But what I hope you can appreciate is there are definitely clumps or clusters, multicellular aggregates of Achromobacter on this epithelial surface. If we do the same experiment with infect a mouse and then recover lung tissue and do immunohistopathology on it, we also see polymicrobial aggregates on the airway mucosal surface following infection, and and that is not strain dependent. Every strain in our clinical isolate set seems to do pretty well at both forming biofilms in vitro and colonizing, persisting, and forming these aggregates in mice, both CF or normal mice. Okay, so if we mix the two on a airway mucosal surface, basically we see the same thing. They are not necessarily as interspersed as we saw in the initial acute infections with the two, but they definitely do form polymicrobial aggregates on this epithelial surface. However, if we quantitate the bacteria, there's no appreciable change. This is over a 6-hour time point. As compared to the inoculum, we see no difference in Achromobacter, whether it's by itself or if it is in co-culture with co-infection with Pseudomonas aeruginosa. In this case, this is a mucoid clinical isolate from our CF clinic. On the right, you will see that in contrast, Pseudomonas counts declined over time with after 6 hours incubation with Achromobacter. So Caitlin next wanted to ask, is this something specific to mucoid strains, since we're using a mucoid strain? And so we used a laboratory isolate strain set that widely used in the field. FRD1 is a very mucoid laboratory isolate, and this is, the one on the right is a non-mucoid derivative by complementing Maquet. And what I hope you can appreciate is that we see the same phenomenon in adherence of FERD1. It declines in the number of viable bacteria decline in the presence of Achromobacter, whereas it does not for the non-mucoid derivative. What does this mean in terms of infection? Basically, we see the same phenotype. And I should say these infections went out we have data out to 96 hours. The complicating factor was the ones infected with Achromobacter, and certainly with Achromobacter and Pseudomonas, had significant mortality that was increased in the polymicrobial infected group. But what you can see is at 48 hours, when we still did have fairly robust experimental groups, there is a significant difference in the Pseudomonas in A recovered from polymicrobial infection as compared to, in polymicrobial infection as compared to animals infected with Pseudomonas alone. On the right are Achromobacter counts, and as we observed in vitro and on epithelial cells, Achromobacter is unaltered. So another thing that we observed is that the inflammatory response seems to be decreased in the polymicrobial infection group, which to me was a little surprising because they did die, but that was what we saw by histopathology and by cytokine measures. They appear to go down in the polymicrobial infection group. So we reasoned that the one possibility, since this phenomenon was specific to mucoid strains, was that Achromobacter xylosoxanins may be able to utilize alginate. We often describe in the biofilm field the biofilm matrix as an edible scaffold, but the number of times that we talk about it being eaten or what eats it is not very many. And so in this case, on the left, we designed a minimal medium. It's an M9 base with alginate in red as the sole carbon source, or in black, glucose. Green is the negative control that has no carbon source. And what I hope you can appreciate is that our Achromobacter xylosoxanins isolate depicted here can grow quite well on alginate, if anything better than on glucose. I should also add that this was reproducible across our entire clinical strain collection. So I think we've tested more than 30 strains. On the right, we next wanted to ask, does this affect something that is clinically relevant? And we thought that resistance to tobramycin killing would certainly fit that bill. What you can see here are Pseudomonas counts. In the black, it's Pseudomonas alone. In the red, it's Pseudomonas with Achromobacter and no antibiotic treatment. And then in the blue is Pseudomonas alone. And then in the green, Pseudomonas plus Achromobacter treated with tobramycin. And our interpretation of this result is that Achromobacter sensitizes Pseudomonas to tobramycin. So our conclusions from these data is that Achromobacter has the capacity to colonize and persist during polymicrobial infection with Pseudomonas aeruginosa. And this is, in previous work from the lab, we actually had shown that Stenotrophomonas has the same capability. In that case, they act cooperatively and not competitively. So it is much more complicated than what I was taught in graduate school, which was that mucoid Pseudomonas takes over and that's the only bug. The antagonistic effect on Pseudomonas populations carries independent of animal model, and it's specific to mucoid strains. So we would, based on the ability of these bugs to utilize alginate, we interpret this as a potential for alginate scavenging or the eating of alginate by the Achromobacter as a mechanism that potentially drives microbial competition during these polymicrobial infections. As I mentioned, the hero of this story is Caitlin Billiot, who has now moved on to a postdoc at St. Jude's. Dr. McDaniel was part of the lab at the time that this project started and was very helpful in its inception. We got clinical isolates from Timothy Starner, who at the time was at University of Wisconsin, and Bill Benjamin, who at the time was the head of our clinical laboratory. And we've also gotten a tremendous amount of help from colleagues in the UAB Center for CF Research, and this was funded by a CF Pilot Exploratory Research Award on Achromobacter. Thank you.
All right, questions. I have a starting question for you. So what do you think is happening with the mice dying? And it's really interesting you saw the reduced inflammation.
Okay, so what we did do, things we have tested that are not the answer. It's not going septic. So there's nothing in neither Pseudomonas or Achromobacter was recovered from the bloodstream. They do have a very serious pneumonia, although, like I say, strangely, some indicators of inflammation, most indicators of inflammation are down. I think they're dying from pneumonia. They certainly have incredibly labored breathing. We did not check for any neurological symptoms, so I guess it's a formal possibility that we could have meningitis or something like that, but I would lean towards they're dying of pneumonia.
I have a question. That was a really great talk. So have you checked to see directly in people with CF if there's an inverse correlation between the abundance of mucoid Pseudomonas and Achromobacter?
It's a great question. And maybe some of if some of my colleagues who are more familiar with clinical epidemiology have something to talk to me about on this, I'd appreciate their instruction. But to me, the existing epidemiologic data don't really give us anything to satisfactorily address that question. Achromobacter is admittedly, it's not in a huge percentage of patients like mucoid Pseudomonas. I will say both Achromobacter and Steno, like non-tuberculous mycobacteria, are in the patients that do the worst. So I don't have an answer for your question. This is really the answer.
And maybe a follow-up question to that. So Barbara yesterday talked about mucoid Staph aureus. Have you done any?
No, and no, we haven't. We haven't. And I must, if you'll permit, we did some work initially on what were initially classified as pneumococci that were isolated from young children with CF. It turns out they're not pneumococci. They are kind of like hybrid strains between orals and pneumococcus, and they overexpress capsule. And I had the darndest time getting that published because people hated me using the term mucoid. So,
okay.
Anyway, no, we haven't done that.
I have a question from Sebastian.
Hi, great talk. So a follow-up question like that. From what I understand, the mucoidy is from these two pathogens. The molecule alginate in Pseudomonas, the chemical structure is very different from the chemical structure for Staph. And in Pseudomonas,
you're right. I didn't go there, but yeah, I think there is a granule of reason why someone would object to the use of the word mucoidy, because what Staph is overexpressing, if I followed the talk correctly, is the ICA extracellular polysaccharide, which is a very different beast than alginate.
Yeah, so
It would have to be a very promiscuous glycosidase.
Exactly. So that is my
that.
question, that you, from your data, you saw that Achromobacter can grow on alginate, like using as a carbon source, if I understand well. So, and the two carbons, the two molecules that made up alginase, hyaluronic acid and hyaluronic acids. So, okay. And that is the, Pseudomonas synthesize that with algae, so Achromobacter should have a enzyme that does the opposite.
I can't satisfactorily answer your question or where I think you're going, but I can give some insight, and it probably would tell you where we're going with this. Just genome gazing, there are two annotated predicted alginate liases that are very highly conserved in Achromobacter psilocybe genomes, and they're preferentially found in clinical isolates as compared to environmental isolates in work that one of my colleagues has done. One of them targets the mannose component, and one of them targets the guanylate component. And I'm working very diligently to knock those out. That's not trivial.
Yeah, that's a good point. Thank you.
All right, thank you, Ed.
Next speaker is Jack Hongel from National Jewish.
Thanks to the organizers for having me, and I can't wait to talk to you today. I'll just jump right on in. I have no relationships to disclose. As Dr. McDaniel did a great intro on non-tuberculous mycobacteria, I just want to repoint out a few things. NTM are opportunistic pathogens that cause lung infections only in susceptible hosts. People with cystic fibrosis are at some of the highest risk for this. The majority of these infections are caused by Mycobacterium abscessus and M. avium complexes. We're going to talk mostly all about M. abscessus today. Treatments are long, limited, and have a high failure rate. And so an increased understanding of NTM's host pathogen interactions are needed for the development of more and better treatments. So let's take a look at what happens when a healthy person inhales NTM. The first line of defense is the mucociliary clearance. Any bugs that make it all the way down to the alveoli are met with the resident alveolar macrophages, which are constantly bathed in a cytokine granulocyte macrophage colony stimulating factor, or GM-CSF. This is produced by the type 2 pneumocytes only found in the alveoli. When somebody with CF or bronchiectasis inhales NTM, by definition, there are some areas of impaired mucociliary clearance and also damage, hyperinflammation, hypermucus production. All this is to say they get mucus plugs in the airways, trapping the bacteria there. And the infections that happen in these airways are dealt with recruited immune cells from the blood. The macrophages from here are matured in macrophage colony stimulating factor, or MCSF. We think that the differences in these two cytokines is important to understanding host susceptibility to non-tuberculous mycobacteria. So we know that GM-CSF is associated with immune control of M. abscessus. Previous studies have shown that GM-CSF knockout mice develop chronic pulmonary M. abscessus infections. Inhaled supplemental GM-CSF showed improved disease outcomes in patients with persistent M. abscessus infections. In some clinical trials, our lab has shown that monocyte-derived macrophages from healthy donors matured in GM-CSF kill M. abscessus better than those matured in M-CSF. The graph that I'm showing you here on the right is those MDMs. We infected them with M. abscessus for 4 days. After which we lysed and plated for CFUs. The y-axis here is showing the amount of bacteria at 4 days compared to what we initially gave them. And as you can see, with higher concentrations of GM-CSF, there's increased bacterial clearance. So this begs the question, though, if NTM pulmonary disease is a matter of insufficient GM-CSF in the airways, then why doesn't everyone with CF get an NTM infection? The current model being, again, GM-CSF only being produced in the alveoli and the airways being a GM-CSF low environment, then everybody should be at the same risk. However, only 10 to 20% of people with CF will get NTM pulmonary disease. So this begs the question, could GM-CSF be present in the airway programming recruited macrophages to kill NTM. So we know that susceptibility to NTM is likely a multi-hit model balancing host factors, mycobacterial factors, and co-infections such as Pseudomonas. As Dr. McDaniel earlier was showing a great data on the direct interactions between Pseudomonas and mycobacteria itself, we're more interested in seeing how the co-infection affects the host factors and how that shifts the balance either to or away from the host, leading to susceptibility. With the host factors and co-infections in mind, we did a literature search. And lo and behold, back in 2002, Dr. Alice Prince showed that Pseudomonas aeruginosa can induce GM-CSF production in CF bronchial epithelial cell lines. Here's just showing different strains of Pseudomonas, PAO1 being wild type, PAO1P being a pillin knockout, PAO-PMM being a defective LPS, and PAO-R1, which lacks quorum sensing. Notably here, only the pillin knockout showed a decrease in GM-CSF. And further in the paper, Dr. Prince showed that the Pseudomonas induction of GM-CSF was dependent on flagella and pili. Years later, a Danish group showed that it was acute, or here they say intermittent, Pseudomonas isolates, but not their chronic counterparts that can induce GM-CSF production in alveolar cell lines. As I'm sure many of you here would know, one of the hallmarks of evolution from acute to chronic Pseudomonas is often associated with the loss of flagella and pili expression. With all that in mind, we came up with a biphasic model of how we think acute versus chronic Pseudomonas isolates may influence susceptibility to M. abscessus in the airways. So, so early on, during an acute infection that still have the flagella and pili, that can induce the epithelial cells in the airway to produce GM-CSF, so then recruited macrophages during that time can be programmed to kill NTM, but after years of infection and loss of flagella and pili, they are no longer able to induce this GM-CSF in the airway. Thus, the NTM survives within the MDMs, leading to infection. To study this, we separated it out into 3 questions. Question 1, can Pseudomonas stimulate GM-CSF production by primary airway epithelial cells in vivo and in vitro? Question 2, how does acute versus chronic Pseudomonas aeruginosa clinical isolates affect GM-CSF production by primary airway epithelial cells? To question 3, does co-infection with Pseudomonas aeruginosa affect host susceptibility to M. abscessus? We're still pretty early on in this study, so the rest of this talk is really going to be focused on question 1. To study question 1, we needed 2 tools. 1 being a mirroring infection, mirroring model of airway infection and not alveolar, and 2, a reporter mouse that shows localized GM-CSF expression. Tool 1 came to us in the form of agar beads embedded with bacteria. The beads themselves are about 100 to 200 micron in diameter and lodge reliably within the small airways. Our labs and others have shown that this agar bead model results in a prolonged and chronic infection compared to bacteria just in suspension. At National Jewish, Dr. Ken Malcolm really mastered and taught us this technique. It is quite difficult, and we're very grateful for that. Tool 2 came to us from the Loxley lab as a transgenic mouse that produces a bicistronic mRNA encoding both GM-CSF and TD tomato. So as the normal expression of GM-CSF is done and secreted, the TD tomato still stays within the GM-CSF producing cells. Here on the right, we have lungs from an unmanipulated mouse from our lab. The nuclei here stained with DAPI and the GM-CSF reporter being here in red. As you can see throughout the alveoli, there's still type 2 pneumocytes all producing GM-CSF, shown by that beautiful spattering of red. These mice come with an extra advantage of having almost their own control, as you can see in that nice large airway down in the corner. None of the epithelial cells themselves are producing GM-CSF, which, as according to previous studies, they shouldn't during healthy. So now that we had our 2 tools, it was time to put it to the test. So we used either heat-killed Pseudomonas aeruginosa with flagella and pili, that's still intact. The live M. abscessus, or just sterile agar beads, inoculated those into a GM-CSF reporter mouse, and after 3 days, we harvested the lungs and processed them for immunofluorescence. To show you what we're doing here, the cell walls are stained with lectin in white, the nuclei again, DAPI in blue, and the GM-CSF reporters are in red. To orient you to what you're seeing here is we have a sterile bead lodged in an airway. You can see that nice inflammatory cell cuff. We also drew in a basement membrane here just to kind of help differentiate those epithelial cells. As you can see in the alveoli, again, there's still those type 2 pneumocytes still producing GM-CSF, but within the airway itself, an infection, not infection, but in the airway, in that spot of inflammation, there's no GM-CSF being reported either in the epithelial cells or the inflammatory cuff. We saw the same phenomenon in M. abscessus agar beads. Again, seeing no red being present anywhere within the epithelial cells there. Further kind of supporting that these M. abscessus mono infections are happening in a GM-CSF low environment. But what about the heat-killed Pseudomonas beads? Here again, you can see where the bead was by that remaining inflammatory cuff. Unfortunately, based on how we process them for immunofluorescence, the beads sometimes lift up. But around the inflammatory cuff itself, again, you can see in the epithelial cells, there's a great robust red GM-CSF producing reporter all around the inflammatory cuff. So we can, it seems like we can say that heat-killed Pseudomonas beads can indeed induce bronchial epithelial cell GM-CSF expression. I also like to note that this is a pretty focal phenomenon here, where as you go farther down in the airway, you lose that any GM-CSF reporter. Now that we had that, now that we showed it in vivo, we kind of wanted to parse out in vitro what's happening just with the epithelial cells themselves, without all the extra noise that happens in vivo. To do that, we collaborated with the Vladar lab at CU Anschutz in Aurora, Colorado, using air-liquid interface cultures to do this. For anyone who doesn't know, they do this by seeding bronchial basal cells, human bronchial basal cells onto transwells, let those proliferate for 5 days, and then they lift off the apical, apical media. So then the cells are exposed to the air and let those differentiate for 21 days, producing a great model with all the airway cell subpopulations. Now that we had our air-liquid interface cultures, we stimulated them with either live Pseudomonas aeruginosa, M. abscessus, or media control. After 24 hours, we collected their apical liquid and determined cytokine concentration. Encouragingly to what we saw in vivo, we saw that in vitro, again, the Pseudomonas aeruginosa, shown here in pink, induced GM-CSF production by primary airway epithelial cells. And also, not only that, we saw that M. abscessus, shown in orange, did not show any significant difference between itself and the media control. Other inflammatory cytokines also followed the same pattern. In a repeat experiment with different donor cells, to importance of science, being able to repeat it, we saw again that Pseudomonas had the same trend where it induced GM-CSF production by primary airway epithelial cells. M. abscessus in orange again didn't. We also stimulated here with just flagella alone to see if that could induce, and whilst modest, there was an induction. So that was again encouraging to what we saw in previous literature. So to answer question 1, if Pseudomonas aeruginosa could stimulate GM-CSF production by primary airway epithelial cells, we say yes. And starting to answer question 2, we're looking into clinical isolates. that we have, and we'll be comparing those in vitro to see if there's differences in the early and late pairs for GM-CSF production. This is all building up to our question 3, which will take beads and put both those either an early or late pair of Pseudomonas with M. abscessus and infect mice with those. And we're predicting that co-infections with the acute clinical isolates will result in lower bacterial M. abscessus burden than co-infections with those of chronic infection isolates. With that, I'd really like to acknowledge everybody who was a part of this, and thank you, and time for questions, I guess.
Thank you. That was a really good talk. Thierry Van from Emory in Georgia Tech. So I really liked some of the visualizations you had. If you could bring back the one where you had Pseudomonas, the Pseudomonas beads, and yeah. So I can see you have also some immune cells in there, large, especially in the corner. Does your reporter work in such a way that if those cells make GM-CSF, they will also light up with your TD tomato?
Yes, they would.
That is, if it's not induced, but if it's actually endogenous?
Yes, the cells themselves would have to produce the GM-CSF themselves.
Okay, and that's because what I'm thinking, and I've been seeing that in patients, patient sputum, we do see immune cells able to actually release some pre-made GM-CSF, some that they made actually, a lot of time earlier in the bone marrow that might have been sort of, packaged and not, and after that the gene is not expressed anymore. So what's your, the question really is, how long does your reporter gene last for? It's like permanent?
With just how delicate the TD tomato reporter can be itself.
Yeah.
It depends. Here we actually use a tyramide signal amplification to show the reporter, get the reporter to actually show just based off of the processing. It takes can sometimes quench the regular TD tomato. I'm unsure for how long the TSA could actually detect the TD tomato after it's been kind of degraded, but that's a good question.
Okay. I mean, one of the options would be to actually visualize GM-CSF itself in addition with the reporter. But it's a pretty beautiful study. And the second question, if I may, downstream of that, you have activation of IL-17. So did you look into sort of the downstream effects of GM-CSF?
Not at this time, but that's a great thing.
A really good job. Thank you.
Thank you.
Right here.
Hey, great talk. I just had a question about the agar bead model, in particular for the Mycobacterium abscessus. And I could be wrong about this, but I was under the impression that part of the Pseudomonas bead model is sort of like release over time, which maybe is partially the motility of the Pseudomonas, like kind of crawling out of the beads. With the Mycobacteria not being modal, and then also if you're planning to do the more chronic isolates that maybe don't have your flagella and pilus, have you checked to see how much contact the epithelium is actually having with the abscessus in that model?
Great question. So for this model itself, they're heat killed, so they.
Yeah, okay.
Yeah, so we still, even though they're heat killed, they can't move at all, we can still see that GM-CSF induction happening in the epithelial cells themselves. But great, we were thinking about that with if it was a live Pseudomonas, if the downstream in those airways, if we would still see GM-CSF reporter with them.
Floating around.
Floating and moving around.
Thanks.
Okay.
Antonio Recchiuti, University of Chieti, Italy. I really like this model, and I have two questions. One is a technical, and the other one is more general. So in this mouse model, you only detect GM-CSF that is made by the epithelial cells, or because this is tissue-specific reporter system, or you can detect other cells producing GM-CSF as well?
We should be able to detect all cells, but here it was, our epithelial cells that produced it.
Okay. And the second question is, I'm not a clinician, excuse me if this is not precise. if I understand you correctly, early clinical isolate Pseudomonas aeruginosa are protective against NTM infections in people with CF. So is this in children with CF get less NTM infection clinically compared to older people?
Just making sure I got your question right. Like, is there a correlation?
Because you said that
early?
I mean, one of your question is if Pseudomonas isolates early in the lungs are because they have expressed flagella, they can induce GM-CSF, which is protective against NTM. So if this is your hypothesis, and clinically, do you see less NTM infection in young people compared to old people with CF?
As I'm not necessarily a clinician myself, so I don't know if I can perfectly answer that question, but we know that, like, as Dr. McDaniel was showing, NTM infections tend to happen for older people who have more progressed disease and lung damage. With there being a correlation of early Pseudomonas and NTM itself, I know that there's been some studies that have kind of shown different results, whether or not there's so much correlation. But if that's a great question.
Well, I mean, congratulations, very good work. And for the next steps, I'm sure you will have good results as well.
Thank you.
Hi, great talk. So what is the role of toll-like receptor 5 here?
We have not yet looked into that, but that I'm sure is a great question. But yes. Sorry.
That's fine. That's my question number one. Question number two, that it's all these, the NTM and all the mycobacterium, always they behave like tumors, they escape the immune response and all of that. So when you talk about tumor persistence, all of this, they are evading the adaptive immune response. And when you're talking about GM-CSF, it comes to my mind antigen presentation and all the evasion of the adaptive immune response. So do you think that there is a role of that you are restituting the ability of antigen-presenting cells to activate T cells when you administer the GM-CSF? And that's the reason why now you're clearing the infection. Yes.
That's also a great question. I'm personally not sure. We've only really been looking into the macrophages for this, with just the macrophage alone, but looking into T cells themselves would be not themselves, but how T cells play a role in this could be interesting.
And out of curiosity only, do we know if there are some T cells dysregulation in patients with MTM infection?
I am unsure of that question at this time. If anybody knows, that'd be.
So I think there's a difference between what's happening in the airway versus once it's infected into the parenchyma. And so the first step is just understanding what's the first reactions with the macrophage. The T cells are not necessarily being recruited into the airway. And so that all of that data, a little bit from MS-sepsis, but more from TB. And so once it's into the parenchyma, then you have the adaptive immune response.
Yeah. So that will be it's a very interesting axis. And also, I will assume based on the basic and canonical immunology that in the presence of TLR5 stimulation, maybe your dendritic cell, whatever is around, will be migrating to the local lymph node to activate T cells, maybe. Yeah. Sorry, Katie. Yeah.
Can I ask a follow-up to the flagellum question? It sounded like you were going to do this, but with your chronic isolates, are you going to throw them on the ALIs and see if you still get the GM-CSF or if it's muted?
Yes. So the plan for question 2 is to look at those early and late isolates on in vitro first to see which ones have which pairs have a large difference, test them for flagella and pili, and then put those into beads.
Yeah, great.
And do you think you could also apply these observations to other microbes, other gram-negatives, or is that specific to Pseudomonas?
You know, eventually I'm pretty sure there's going to be a grant coming in soon for looking into other pathogens.
Great. Thanks so much, Jack, for your talk. It was really great.
Thank you.
All right, last up, we have Anna Galdino.
Good morning, everyone. My name is Anna. I'm a project scientist at Peter Jorf Lab at Cedars-Sinai. But today I'm going to present a collaboration work with Liz Bergener from CHLA. And today we're going to talk about how the CFTR modulators affect both the microbiome and the phagome. And before I start my presentation, I would like to apologize in advance. Today, when I arrived here and checked my presentation, I realized that I had one of those Mac PC incompatibilities, so my images are quite blurry. But I'm going to try my best to make them a little bit clear. So, as we know, one of the consequences of the CFTR mutation is the impairment of the mucociliary clearance and the accumulation of mucus, which predispose people with CF to acquire chronic infections and favors the several microbes to proliferate and form a complex and diverse microbiome. And among the virus who compose the CF microbiome, bacteriophages are the most prevalent in the CF sputum, and they have a key role shaping the microbial community, either by killing the susceptible bacteria or by conferring fitness advance in some specific lineages. And more specifically, the filamentous phages who infect Pseudomonas aeruginosa are highly prevalent in people with CF. So previously in this study, it was identified that in this CF Danish cohort, around 70% of the Pseudomonas isolates who colonize those patients carry genes of these filamentous phages. Also, those filamentous phages are correlated with a more rapidly decrease in lung function, which highlights the relevance of phages and the phage composition in the disease progression. So this is a good reason to better understand the composition of the phages within the lungs. So with the CFTR correction, with the CFTR correctors, we've been observing an improvement in the clinical outcomes, an improvement in the lung function and the sweat chloride. And initially, we thought that with the improvement of the lung environment, we would also have an improvement in the microbial parameters. However, this is not what we've been seeing. Here, I'm showing a previous study where it shows, although we have a decrease in Pseudomonas burden, Pseudomonas sticks around for even after 3.5 years after ETI initiation. And this same pattern is being observed by 2 other CF pathogens. And although we already know we already have a good idea how the microbiome reacts after ETI initiation, we have no idea how phagome is affected by the CFTR modulators. So here in this study, we analyze how the modulators affect both the microbiome and the phagome. And for that, we have analyzed 63 sputum samples from 51 individuals with CF. Of those, we had 19 who were receiving the modulators and 44 who were not receiving. And among those patients, we had 12 paired samples, which was pre and post ETI. And in our cohort, we have a wide range of age, ranging from 8 years old to 66 years old. And they also had a wide range of disease severity levels. And for this, we also, once we had the sputum, we did the culture, we checked the Pseudomonas and the phage quantification by qPCR. We performed a shotgun metagenomics analysis to identify the microbiome and the phagome. And we measured the active neutrophil elastase as a proxy for inflammation. And for the analysis, we compared the data using two different approaches. One is comparing the samples non-paired. So we would capture the difference between these two groups. And we also compared the paired samples to capture differences within subjects. So initially, as we already know, the CFTR modulators can improve the lung function. Also, we saw that it can decrease the neutrophil elastase activity, so it's improving the inflammation. And this improvement in the inflammation status is correlated with, it's inversely correlated with the lung function improvement. And looking at the microbiome, we saw that most of our samples here represent, most of our samples were dominated by Pseudomonas aeruginosa, here represented by the green bars. Two samples become Pseudomonas positive after the ETI initiation. And one sample were negative pre and post ETI. And only one sample managed to clear the Pseudomonas infection after ETI initiation. And when we quantify the Pseudomonas burden, we could confirm that Pseudomonas persist after ETI initiation. And the same pattern was observed when we compared the two groups, non-pair groups. Pseudomonas persist, they have similar burden, but however, we saw that we had an increase on microbial diversity, which may indicate an improvement on the microbial parameters. Interestingly, we saw Staphylococcus were decreased in abundance on the microbiome, and we saw that we had a decrease on the proportion of methicillin-resistant Staphylococcus aureus. On the other hand, we saw an increase in abundance of Streptococcus on the group who were receiving the modulators. And when it could, because Streptococcus are commonly associated, commonly considered as a commensal, they might indicate this increase in the Streptococcus abundance might indicate a more healthy microbial community. So now looking to the phage part of the microbiome, we didn't see any huge difference on the phage structure when it comes to the phage family, and there was no difference on phage diversity as well. And looking at the Pseudomonas infecting phages, so they follow the same trend as the host, the bacterial host. So the Pseudomonas phage also persists in high prevalence on the post-ETI group. However, when we look specifically to the Pseudomonas non-aeruginosa phages, we saw an increase, an enrichment of those phages, which may indicate less pathogenic phages colonizing the lungs. Also, we saw a decrease on the ETI we saw a decrease on the filamentous phages burden in the group who were post-ETI, which indicates that the Pseudomonas who were infecting those individuals are probably less pathogenic. And we also saw that the elastase, the neutrophil elastase, correlate inversely with the microbial and the phage diversity, and directly correlate with Pseudomonas and its infecting phage. However, it inversely correlates with Streptococcus and its infecting phage. This study has some caveats. So all of our samples were sputum samples, which biased our population towards a more severe lung disease. However, it guarantees that our samples represent well the lower airways. This is a single center cohort, so it limits our generalizations. And for the phage, some phages were unannotated, which kind of limit the conclusion we can make based on their functional roles and interaction with its bacterial hosts. And our sputum collections was not standardized to fixed times pre and post ETI. So it increases the heterogeneity in our group. Although, despite those caveats, we could conclude that the CFTR modulators did not significantly change the Pseudomonas prevalence in the CF airways. The correlators was associated with the reduced Staphylococcus abundance and increased Streptococcus abundance. And the correlators was also associated with a decrease on the filamentous phages, indicating that decrease in the prevalence of pathogenic Pseudomonas aeruginosa. And we had an increase in Streptococcus and its infecting phage, correlating with the decreased inflammation. So overall, we could conclude that the CFTR modulators create a more balanced microbial environment despite the Pseudomonas persistence, potentially contributed to improved airway health in individuals with CF. And with that, I would like to thank Liz Bergener for this amazing opportunity, my mentor, Peter Jorf, and the Stanford team for the great collaboration. I know we are in the very last day, but if you are curious to know more about it, I'm at poster 66. And if you want to know more how we can improve antibiotic treatment to increase cephiduroccal killing against Pseudomonas aeruginosa, I also have a poster on number 69. Thank you.
Hi, great, thanks for the talk. I just wanted to ask a couple of clarifying questions. So what species were the Streptococcus? I assume it was like a mix, but were there any pathogenic ones in there, or were they all sort of more commensal?
Good question. This is something that I am analyzing right now. So initially, I just look at the genera. I didn't dip into the species, but it's something that I will definitely look at.
Okay, thank you. And then I wanted to ask about the Pseudomonas non-aeruginosa phages. Are they what are they infecting if they're not aeruginosa but also Pseudomonas, or are they just maybe like unclassified?
Yeah, maybe they are. You know, most of the non-aeruginosas that I got was fluorescence and more environmental Pseudomonas. Okay. What they are infecting, I'm not quite sure.
So you didn't see fluorescence, the bacteria in the sequencing?
No.
Okay, cool. Thank you.
I don't need that. Great job. I was wondering if you do have the ability in any way to look or correlate with things like lung function and time. And the only reason I ask is because an FEV1 of 25 to a range of 125 is a very different lung environment and a very different person. And I think that can be beneficial here, and that maybe, and I know it's a numbers game at this point in time, but maybe you're seeing more pattern of these shifts and changes that may have less or more effect with modulator pre and post, which is a great way to do it. But, we're talking somebody who has a quarter lung function compared to somebody who has, 1.25, if that makes sense. And so along with that time, I think you could potentially, or if you have any plans to, map that out, or any other clinical things that are going on, knowing who you work with and things, so she can do that. That's all.
Yeah, it's definitely something that we discuss a lot while we were working on this data set. But surprisingly, when we were doing the correlations with our samples, we were expecting to have a bigger role on the lung function, and it was not that clear. But it's definitely something that Liz will follow up in the future. Yeah.
Yeah, hi. Thank you for the nice talk. I was just curious, like in your genomics analysis, do you also see changes in the fungal environment?
Yeah, I didn't
and whether your changes in the bacterial community is the direct outcome of the HEMT therapy, or overall changes in the fungal environment and then contributing towards the changes in the bacterial community?
Yeah, I focus more in the bacteria, in the microbiome. I didn't look into fungi. For your second question, can you remind it?
Yeah, for example, like I was curious that whether the changes in the bacterial community or the population changes which you see is the outcome of HEMT therapy, or is it the outcome of changes in the fungal environment, like the colonization of the fungal species, and that is leading to the changes in the...
Yeah, I cannot answer your second question because I didn't measure the fungal community at all. So, yeah, I don't know the answer for that. I'm sorry.
I have a question for you, Anna. That was great talk. So I'm just always amazed when you see their changes post versus pre-ETI. So I'm just wondering, in some of those patients where you haven't seen changes or where you've seen big changes in community composition, can you just go back earlier in time to see if you have access to sputum samples and see if this tracks these individuals who had big changes were more likely to have a community that would change or would be more stable?
Yeah, honestly, I don't know if we had access to those sputum samples for those patients. Yeah, I don't have the answer for that.
Can I ask one more question? Can I ask you to speculate on what you think is happening with these correlations with the neutrophil elastase signal?
Yeah, for the Pseudomonas, it's already known that Pseudomonas is highly correlated with the neutrophil elastase. So it's kind of intuitive to speculate that an increase on the Pseudomonas correlates with the neutrophil elastase. So if Pseudomonas is there, probably Pseudomonas phage increase would also correlate with increased elastase activity.
Yeah, I guess, is there any phage specific signal you're picking up, or is it all which bacterial host is there?
Yeah, I didn't test against specific phages. So most microrelations were focused on the host the host of the phages, basically.
One other question. So do you know if total phage concentration has changed?
Did you test that? Did you measure? Just PF, yeah.
We're curious to see if it's a matter of like the ratios, or if there's more phages or less phages?
Yeah, that's a good question. We didn't check that.
All right. Thank you, Anna. And thank you to all of our speakers for a great session.